Welcome to the study of sensation and perception. Sensation and perception was the earliest area that psychologists studied. Before this, psychologists were philosophers, and psychologists were philosophers who examined mental processes like reasoning and memory and things like that. But when psychology transitioned from philosophy to become a science, the first thing that it used the scientific method with was sensation and perception. So the first empirical studies in psychology involved cutaneous sensitivity, sensitivity of light perception, hearing, and things like that. So the first psychology laboratories were devoted to trying to figure out what the parameters were for sensory experiences. What kind of light level did you need to perceive a light? What kind of low level of sound did you need to perceive a sound? What was the softest sound you could perceive? And then issues of like, well, how much does a light have to change before we notice that it's changing? These things were considered fascinating to study at the time because before this, all mental operations were considered a kind of spiritual entity, a non-material thing. And here the psychologists were making the mental functions very material and making them something that could be studied scientifically. And that was quite a novelty at the time. And it really launched psychology as a very sort of special discipline of the world. We were the first ones to examine, in a scientific way, things that are examined today all the time from scientific method. Well, sensory experiences represent the input into your central nervous system. They represent the information about the world, information about the environment, impinging on your senses, which are specialized receptors for interpreting that kind of information. Then an encoded form of the sensory experience is sent up into your brain, and you formulate a perception. You formulate plans for acting on the environment. You figure out, well, the apples are red, that means they're ripe. When they're green, that means they're not ripe. And you decide, okay, today's the day we collect apples because they seem to be ripe today. Well, the sensory processes that you went through to make that decision all involve vision, all involve sensitive systems that detect different light levels, different light frequencies. You formulate a perception of a red apple, and you've had experiences with red apples being ready to eat, and so you go out and you collect them. Well, you could imagine all these sensory systems working together to produce essentially a picture of the world that you act on. And without that information, your environmental adaptation would essentially be very poor. Well, because these things involve stimulation, the things that the entity of unit or of examination for sensory experiences is the stimulus, and stimuli for the plural. A stimulus is defined pretty broadly in this area, is essentially any sort of unit that can be identified that impinges on a sensory system. And so a stimulus can be a full picture, a full visual array, that could be considered a stimulus, or a single sound presented to your hearing system might be a stimulus, or a pinprick on different parts of your skin for cutaneous senses. All these things are referred to as stimuli, but again, they're defined quite broadly. So stimuli can be any kind of unit that's involved in that study at that time. There's a strong contrast between sensations and perceptions. As you might imagine, your sensations are the initial impact of the sense of the physical environment, say physical energy like in the form of light on your light receptors, or a sound wave onto your hearing receptors. These are your sensations. Now your perceptions are reconstructions of sensations. So your perceptions are essentially how your brain has taken that information about sensation and mapped it as a perception. A very good example of this is color perception. Now colors, you'll be perhaps surprised to know, maybe you know this already, colors do not exist in the world. The color blue is not a property of the world. Blueness is not out there as a property of an object. What objects have are surfaces that reflect certain frequencies of light. And we have receptors in our retina of our eyes that respond to those frequencies, and send an encoded pattern up into the brain. Your brain visual systems, which take up about the back third of your whole brain, then process this information and map the world by colors. So blueness, redness, yellowness, all these things do not exist in the world. They're reconstructions of the sensation so that we can make discriminations in the world based upon light frequency information, which your brain has interpreted as colors, interpreted for you as colors. And then when you operate on the world, you operate on colored things. But color itself is a perception. The light frequency information impinging on your retina is your sensation. So the perception is very different than sensation when it comes to color. Most of the time, there is a pretty close matchup of the sensory experience with the perception. The perceptions might just represent actually a kind of enhancement of sensation so that you can discriminate better. Sometimes they're illusions. Sometimes perceptions are inaccurate representations of the world. Psychologists have examined these and have collected these. Fortunately, these illusions are quite rare, and they involve essentially some sort of strange sensory perception that a psychologist has to construct for you to be able to miss a sensation, have an illusion. In the real world, in the real environment, they don't happen very often, and they usually don't involve anything that would really change how you adapt to the world. Fortunately, we're all quite similar in our perceptions, all quite similar in our sensations. It's amazing that all these different nervous systems developed and matured in much the same way, giving everybody the same sensations of color, pretty much, same sensations of hearing and things like that. Now, there are differences among us. There are some people that see colors better than we do, than most of us do. There's some people that perceive sound better than others do. But we're remarkably similar. I mean, when somebody says, here's a red apple, we pretty much all see the apple the same way. Some people see it slightly different red color than others do. But the mapping of color onto frequency is very, very similar among us. And so there's a high degree of reliability. I think to the point where, most of us don't really think about color not being in the world. That's a novelty as a concept to most people, because when we talk about colors to each other, we're also consistent and reliable in how we talk about these things, that we don't recognize that they're actually in our brains, they're actually in our mind, and not actually a property of the environment. Okay, the next concept here is that of a receptor. I've already mentioned receptors. Receptors are specialized cells in the sensory systems that take that physical information, the physical forces or light energy or a sound wave from the air, and they take that physical object from the environment, a physical force from the environment, and convert it into a neural firing pattern. And the process in your book and other places you'll hear about this is called transduction, this conversion of a physical force into an encoding pattern in the nervous system. It's not quite transduction in the sense that physicists refer to transduction as the conversion of one energy form into another. Essentially, the nervous system works in a way that's much more similar to a analog digital converter. So when I'm sitting here recording these lectures, I'm talking into a computer that takes the sound wave of my voice and converts it into data, into numbers over the course of time that reflect that sound wave. And depending upon how accurately and how often it takes a reading will determine how accurate and smooth my voice will sound. Well, that data that it's being converted into is something similar to the way the neural system, the neuron firing patterns, represent an encoded version of the sensory stimulus, and in much the same way. And these encoded patterns have yet to be discovered in any sensory system with great accuracy. Obviously, the neurons are firing in some kind of pattern that the brain can interpret as a sound or colors and things like that. But it's more like a analog to digital converter than the other way around, than a transduction system. All these sensory systems use pathways, and here you see the pathway in the central nervous system for hearing in this slide. So the specialized receptors in your ear that interpret the sound wave as an encoded firing pattern of neurons have to convey that through central nervous system pathways that go up into your brain. There's a receiving area for sound information in the temporal lobe of the brain, and everything's laid out in the hearing system by frequency of sound, and we'll get to that when we get to the hearing section. But basically, everything's got to get from your sensory systems into your brain. These are all, your sensory systems. I think a lot of us identify with them by the receptor part of the system. So we tend to see vision as a property of our eyes, as hearing is just a property of our ears, and so on. And we don't really identify with them so much as being a property of the central nervous system. It's the way your brain is getting information about the outside world in order to construct perceptions of it, in order to construct a model of it so that you can act on it. And all these things are brain systems. The sensory systems are all part of the input into the central nervous system so that you can operate in the world in a very sophisticated way. Another distinction that's made is between primary processing of sensory information and secondary processing. The primary processing mostly refers to sensations. So there's a primary processing of the information at the receptor site that turns into an encoding pattern of a neuron, and that's primary processing. And then secondary processing can refer to everything else that the central nervous system does with the sensory information. So if I want to, if I'm out in the woods and I hear a sound of a bird, I'll orient my visual system to the sound source. So the first thing that happens is as I hear the sound, and I localize the sound using secondary processing of sound, and then I turn and orient my visual system to try to locate the object in visual space. And so my brain is working on all the sensory information that's auditory and visual and integrating it into a search pattern for a bird, that's out in the woods that I want to look at, that starts with the sound of the bird. Well, that higher order processing that occurs to integrate hearing and sound has to happen in your brain. It has to happen in your central nervous system, and it's all a part of the secondary processing. Okay, now we're going to go through these in a little more detail, and we're going to introduce some other ideas that are general in this area of the study of perception, of sensory sensation and perception. These are general concepts in this first section that will be applied to every other part of the course. So stimuli, receptors, transduction, all these primary processing, all these kinds of things will be applied to every major sensory system as we go through it in the rest of the course. Here are some more distinctions, some more concepts to go over. The first of these that's emphasized in your book is the contrast between the environmental stimuli versus the attended stimuli. And that, of course, is an obvious, important distinction to make. All kinds of sounds impinge on our hearing system. All kinds of visual information, appears on our retina. But we don't remember it all. We don't process it all. We don't pay attention to it all. There are certain things that we attend to and process and perceive, and then there's other things that we don't attend to at all. And there's a great phenomenon called, I think it's called sight blindness, and it's associated with a movie in this section. And you can watch the movie and get a full appreciation for what we attend to versus everything that we see. It's a great example of how attention influences our perceptions and our judgments. And next below this, we have the five senses. I'm sure you've heard that, before, that there are five senses. Well, get that out of your head. There are many more senses besides just five. There are other ways of breaking down the system except by these five. Now, there are five in the sense that there's sort of five really big ones. But you may, for example, not know that right now, while you're sitting listening to this, your digestive system is sensing whether there's food in your system or not. Your brain is sensing the temperature of your blood. It's also sensing the level of glucose and amino acids in your blood. And whenever those go down, you feel hungry. When your oxygen level Actually, your brain is not sensitive to your oxygen level. Your brain is only sensitive to your carbon dioxide level. So fortunately, they're correlated. They're highly correlated. As you inhale and exhale, you're inhaling oxygen and exhaling carbon dioxide. If the carbon dioxide level goes up, though, that means you haven't inhaled recently. And so your brain wants you to breathe, and you get that compulsion to breathe. You can trick it. If you just exhale, you'll reduce your carbon dioxide level, and your motivation to inhale, your motivation to breathe will go down. You won't have to feel like you need to breathe so much. It's a trick that scuba divers know to use just once. You can't keep doing that. Obviously, you'll fall unconscious. But if you want to reduce your desire to breathe, then exhale. You won't lose much oxygen when you do that. You'll exhale a bunch of carbon dioxide. Your brain will sense that, and you won't have that sort of panicky sensation to inhale. Obviously, you need to inhale eventually, but you can get away with a few more seconds of lower motivation to breathe if you do that. The point is that your brain is sensing all kinds of things outside of these five senses. And it's important to keep in mind these are just sort of a rough rubric to sort of separate these. Well, they're separated into the ones you're mostly familiar with. The somatosensory senses that involve touch are much more complicated than most people understand. Vision, people understand vision. Vision is the big one for primates. Primate vision is the most developed visual system ever in the course of evolution. And primates are excellent seers, excellent viewers of the world. And they use vision for just about everything. Hearing is next. Hearing for primates like us is very important. It's not as important as vision. Clearly, blind people have a much harder ability to interact with the environment as people who are just deaf. Obviously, you don't want to be deaf. You do lose a lot when you lose your hearing. Hearing, though, is sort of secondary to primates. Primates want to see everything. They want to process things visually. They want to orient their bodies in space, and you can't do that very well if your vision is impaired. Hearing is very important to us. Hearing has become obviously really, really important to human primates because we use hearing for communication. And communication allows us to be such well-developed social organisms. It allows us to be able to transmit information to each other, but also to transmit information across generations through the form of writing. Okay, so hearing is very important for us. Somatosensory sensations are also very important. These involve all the receptors in your skin, but also receptors in your joints and receptors in your muscles. You may not be aware that you have receptors in your muscles and receptors in your joints, but every time you move your body, you move your elbow, you move your joints, there are sensory systems inside those joints that tell your central nervous system where your body parts are and how they've moved. This is obviously very important for navigating in the world, for acting on the world. You need to know where your limbs are, and you have sensory systems within them to detect that. Most of us think of touch as the physical force is laid on skin that displaces it, and we see that as touch. But obviously, there's also pain receptors in your skin, and your joints and your muscles. There's temperature receptors in your skin. There's a set of touch sensations, touch receptors in your skin that do all that touch sensation. And finally, you have the two chemical senses, taste and smell. They're called chemical because external chemicals from the environment impinge on the receptors, and you sense them. You sense these chemicals. Now, you don't actually, in humans, taste and smell are very poorly developed. They probably were more developed when we were primitive mammals, but as soon as we got up off our front legs, got up on our hind legs, we got away from all the chemicals that a dog or other animals will sense because they're close to the ground. That's where all the chemicals are. Many of them, of course, are up in the air, but not anything like they are on the ground or near the ground. So we don't use them so much anymore. They've become kind of vestigial almost in humans. There are many people in the world, for example, who have a congenital disorder that made them unable to smell. And some of those people didn't know that until they were adults. They never knew that they weren't smelling. It wasn't like it was a crucial thing to discriminate, objects and so on. You can tell an apple from looking at it. You don't need to smell it. Obviously, smell is an important sensation when you have it, but it's obvious that for humans, smell has become far less important than it is for animals that are close to the ground. Taste also is something you can live without and still survive. It's obviously very helpful to be able to taste things. You can tell if an apple's ripe or too ripe, if you can taste it, and that can help you survive. Obviously, taste is very important for some other animals. For humans, though, pretty much we get through the world by vision, and so we discriminate objects by vision. We don't use so much taste, and many people have lost their taste sensation for a whole variety of reasons, and they don't have as rich a sensory world as they had beforehand, but it usually doesn't affect their environmental adaptation very much. The final general concept on this slide deals with bottom-up versus top-down perception. And this is kind of an odd, it's almost an arbitrary kind of distinction. It has to do a lot with primary and secondary processing. I think you could look at that the same way. Bottom-up processing has to do with how the sensory receptors influence a perception, and top-down processing has to do with how secondary processing influences perception. And the next slide, we have an example of top-down processing that I think will clarify that concept. Here we see an example of top-down processing. Now you notice this is a slide here that's got a picture on the right. Right now what you're seeing is a sensation of, a white block there, a white display, and it looks like there's smudges on it. And maybe you can tell that there might be something written there, maybe not. The fact that I've just told you that it's something written may begin to involve the secondary processing that's going to figure it out. Because now your brain is trying to figure out, you said it was written. That must be some kind of writing there. I'm going to try to figure out what the letters are. And the top-down refers to the central nervous system working top-down toward the sensation to try to make meaning out of it, to try to perceive it. Now as you go from slide to slide, it'll begin to reveal what's there, and your brain should begin to process it pretty quickly as language, and then it's a matter of trying to figure out what the words are, if they mean anything. So go through these slides up to slide 8 there, and you will see it eventually clarify into a set of words and non-words. You see there's some nonsense collection there of letters, and you should have been able to see the true words sooner than the others, because your language centers will process that information, interpret it as writing, interpret it as a graphic form of the sound elements that make up language, and you should have perceived it. Well, clearly that process is top-down. The language centers are in your brain. They're not sensory systems out in the periphery, not sensory systems out there that are just detecting some change in light levels out there. They're systems that process those changes and detect language-related content. Now you should be past the top-down processing slides, on to slide 9 here, which shows this guy holding some weights. One of the first things that the early psychophysics psychologists did And the general area of sensation perception was referred to as psychophysics back in the old days. Still today, I think that terminology is used, even though the courses are usually called sensation and perception. Psychophysics is the study of psychological physics in the sense of the physical properties of the mind, the physical properties of sensation and perception. Well, the first methods to be used involved these things called phenomenology and introspection. Phenomenology is a philosophical point of view that stresses description of what's observable. And so phenomenologists describe the world as opposed to trying to explain it using a number of abstract or internal constructs. I know that doesn't make a lot of sense now, but in terms of the psychophysics, it refers to observation, observing yourself, observing the world, writing up your observations, rather than trying to interpret them and explain them through constructs. Introspection is a methodology by which you use your own internal observing systems, and I don't know how you introspect without sensation and perception. Clearly there was a sense of this observing mind that would introspect. And so when you're going through a research study in which you're comparing different sensory, say different weights of objects like you see here, or different, say an experiment that involved how far apart 2 points have to be before you detect them as different on your skin. That was a common experiment that was done in the past. And rather than measure these things and have people estimate whether there were 2 points they were being touched with or one, the experimenter or the subject and the subject, whoever was being pricked by the pins, would introspect and begin to observe and reason, describe their own perception or their own internal sensations, as well as a way of revealing to the psychologist what the sensations and perceptions were. And introspection involved this self-reflection, this internal look at what the sensory experience was to help define it. Introspection didn't really last too long in psychology. The major proponents of it were Titchener here in America and Wundt in Germany. And Titchener used it a lot. It was basically a way of having the subject describe what they were experiencing, and then the psychologist would make inferences about what they thought that report meant. Okay, well, the early psychophysics studies involved some pretty dry examinations of sensory perception and how some pretty, they were quite reductionistic. They're still used today, though. Still, people, when they're doing sensation and perception experiments, use the same methods as these guys developed. And what they were basically after was they were trying to figure out the parameters of sensation. So they were trying to figure out, okay, what's the least amount of light you need to see to say that you've seen a light? What's the lower limit on the visual system? What's the low limit on the sound system? And when things change, how much do they have to change before you perceive them as different? Well, they developed all kinds of cool techniques for examining this. The first of these is referred to as the method of limits. The second is adjustment, constant stimuli. They're associated with Fechner. I'll go into some of those in a little more detail. And difference detection methods were identified mostly with Weber. He worked on models trying to understand when stimuli change, how much change do we need to have before we perceive they're different. And we'll go into that in more detail in the next slide. Here's the method of limits. In the method of limits, what you're trying to figure out usually is minimum stimuli characteristics that the system will respond to. So what's the lowest light level that the person sees? What's the lowest sound level? And you can see in this graph here in this demonstration of it, there's an intensity measurement here. It's an intensity of a stimulus. So here we've got 105, 104, going down to 95. And what you do is you alternate going from the top, from higher numbers down to lower, till the person says they don't see it anymore. And then you go from lower to higher till the person says they see it. And you can see here the first trial, indicated by the one in the circle there. They started at 105. Person said, yes, I see it. 104, they said yes. 103, they said yes. They got down to 99, and they said yes. Then when they get to 98, the person says, I don't see it anymore. And so they stop presenting stimuli. Then on the next trial, they start with intensity level 95, which was lower than the 98, where they said they didn't see it anymore. At 95, they did not see it. At 96, they did not see it. At 97, they did not see it. At 98, they did not see it. At 99, they did not see it. And finally, at 100, they said, yes, I see it. And so they stopped. Now, notice that before, at 99, they said, yes, they saw it. Where here at 99, they said, no. If you go across the trials, you can see where they start at the high levels of intensity, go down to the lower ones. The next trial will be from the low to the high. You notice that around the band of 99, 98, people say they see it most of the time. Now, that turns out to be the method of limits for determining the threshold of that stimulus. Now, notice that it's not perfectly at 99 or perfectly at 98. It indicates that the threshold levels have some uncertainty in them. There's some error involved. And so it's approximately 98, but it's not exactly 98. And that is an interesting aspect, I think, of biological systems that had to evolve to do this kind of detection, as opposed to the modern devices and machines that we use to do the same kind of detection that are far more accurate than our eyes are. And they've been able to develop devices that enhance our vision with much more accuracy than the actual system. And that's because the biological system, that's being designed by evolution is going to have to live with whatever, you start with, and whatever the constraints are for a biological mediation of this, as opposed to something electronic, something that an engineer would just pull off the shelf to measure light levels. In the old days, when people took photographs with non-digital cameras, they had to go measure light levels, and these devices measuring light levels were obviously going to be much more accurate than our human visual system is. However, we're not that bad. When it comes to visual systems measuring light levels, we're actually quite good compared to other animals, certainly, and quite good in terms of environmental adaptation. But that's the method of limits. And so you can see how that's demonstrated. You start with high or low, and you go to low. You go back to high, go back to low, go back to high, go back to low. And then what you do end up in the end is a set of threshold levels that represent the absolute threshold, in this case, for a stimulus. The next psychophysical method is referred to as the method of constant stimuli. Now here, instead of you going through a systematic, start with the high stimulus, going down to the low, go from the low to the high, like we saw with the method of limits, here you present samples of stimuli and of different levels, and you present them in random order. And so you just present 150s, 200s, 180s, and so on, and you ask the person if they see it or not. Well, the point at which they reach a 50% criterion, in which they're seeing it half the time, and of course not seeing it half the time, that becomes your cutoff for the absolute threshold. And this way you can present stimuli, any way you want to, and you get a nice curve here representing whether they can see the stimulus or not. And at the 50% mark, you arbitrarily, or you just you declare that to be the absolute threshold, because that's the point at which they're making half perceptions and half errors. The final general method for psychophysics I'll be presenting in this section is referred to as the method of adjustment. And imagine this, where stimuli are adjusted by the subject until the stimulus is perceived. And so I put a little dial in front of the subject, and they turn it up until they see the light, or they turn it down until they don't see it. And all the while, I'm measuring where the dial is, measuring how they're adjusting it. And I get a very quick estimate of their ability to perceive the stimulus. Well, the method of constant stimuli is generally considered the most accurate. That's where you're presenting sets of items randomly and choosing the 50% cutoff. That turns out to be better, I think, it's because it's probably not biased in the way some of the others are. The method of adjustment and the method of limits are actually quite similar in the sense that you're moving the stimulus, high and low, and having the person make a judgment about whether they see it or not. Unfortunately, in a situation like that, you have something referred to as priming effects and anchoring effects. And so if somebody says they saw it before, they know it's coming, they know it's there, and so they might say they see it when they don't. Or because they're uncertain about whether they're seeing something, they'll say they don't see something when actually they do. And you end up with a little more error with those methods than when you use the method of constant stimuli. They're all quite good, though. They all pretty much get difference thresholds and absolute thresholds pretty well. It's just the method of constant stimuli is just a little bit better. Now, in the early days of psychophysics, these guys found out some very interesting things about how we make judgments of things like weight measurements and things like that. And one of the things they discovered was the difference threshold. They really worked hard on this, and they tried to find out what's the smallest difference between two stimuli that can be detected. So here you've got two stimuli. You've got the guy holding a plate there with a weight on it, and then you've got him holding a plate with his other hand with another weight. And it was figured out that using the the method of limits and things like that, that the difference limit increases as a systematic function called Weber's law. And Weber's law indicates that there's a Weber fraction or a constant that reflects the degree to which something has to change before we perceive it as different. And here it is illustrated with the guy holding the weights here. Notice in the upper graphic he's holding 100 grams in his right hand and 100 grams plus 5 grams, 105 grams, in his left hand. Now that's just enough percentage-wise for him to perceive a change. And so the difference limit turns out to be 5 grams in that situation. Now give him a 200-gram weight and a 200-gram weight on the left side. Well, here you've got to add 10 grams before he perceives a difference, which is twice as much as he perceived different in the upper one. So the difference threshold is related to the original stimulus condition. So the intensity level of the stimuli, and what you need is a fraction of 5% to perceive a difference, no matter how much is in an absolute sense is changing. So if it doesn't change by 5%, we won't see it as different. We'll still see it as the same. So it doesn't have to change from 100 grams to 5 grams, just to 5 grams, and then from 200 grams to 205 grams. You would think that if he detected a 5-gram change in the top, he'd detect another 5-gram change in the bottom. But that's not what happens. You need a 5% change. So it has to go up to 10 grams if you have a 200-gram weight, and then you perceive there's a difference. Well, no one really expected this kind of difference threshold to exist. It was based upon percentage. I think people expected that there would be a difference threshold that was based on the absolute level of the stimulus. Well, Weber and the others discovered this interesting phenomenon, and it probably has something to do with the secondary processing of stimuli like weight and the way in which your brain processes differences. And it wants to see percentage differences to make a contrast as opposed to absolute levels. And so Weber's law has been applied to a lot of different stimuli, and it's fascinating that it holds up so well given all the different stimulus conditions in which it's been applied. And we'll talk about more of these as we go along through the course. Another method to measure difference is to take a standard stimulus and assign new stimuli as plus or minus in relation to the standard stimulus. So you compare a new stimulus to a standard and assign a numerical value to it. So you decide, well, this light I'm looking at now is 3 times brighter than the comparison light. And so you assign a value to it. Well, Stevens is associated with this method, and Stevens, they refer to these as power functions. Power functions refer to curvilinear functions. They're just not linear relationships. And here you see some Stevens power functions for different kinds of stimuli. Now let's look at apparent length there. That's the one that's in the dotted line. Notice that apparent length of an object, and what you're doing is you say you have a stick and you're in front of you that's 2 inches long, and you're shown pictures of sticks at various lengths, or you're given more sticks to compare, and you make a comparison of the 2-inch length to whatever you're shown, either it's shorter or longer. Well, this tells us that that kind of judgment isn't done in a linear fashion. Pretty much as the stick gets longer, you perceive its length to be greater, consistent with how it's being expanded or contracted. And so your perception is pretty accurate. Now, these other things are showing us that these perceptions are not completely accurate for, say, brightness. As you're shown brighter or softer light levels, for example, for brightness, you make judgments about brightness that's, it's roughly linear. It's clearly nonlinear. It's curvature, but your brightness levels go up consistently with the stimulus of actual values going up. But notice that it's not linear like length designations. So measuring size, we do much more accurately than when we measure brightness levels of a light stimulus. Now look at electric shock. What this is telling us is that as the stimulus intensity increases, our sense of magnitude estimation goes up by exponentially. So as the electric shock goes from a 20 to a 30, we really perceive a much bigger change than 10 units. Then as it goes from 20 to 40, it's perceived as being off the scale. And it's so far off the scale that we don't even have part of the scale left to describe it in terms of stimulus intensity. And if you think about it, it goes along with what we perceive. If you were to, be rigged up to some wires and people were administering electric shock, very soon, every shock you would experience, every electric shock you experience, would just feel as intense as the other one, no matter how much higher the stimulus intensity was. It's just a god-awful shock. We don't have the ability to make fine discriminations in electric shock. As soon as it reaches a certain magnitude, it becomes, just impossible to tolerate. And all the shocks after that, no matter how high they are all perceived as the same ungodly high shock. Temperature is like this. Other sound loudness has some properties like this. Anyway, Stevens' magnitude estimation methods were able to map out these kinds of functions, which are very interesting to compare and contrast. I clearly see, even in this graphic, is telling me that vision for primates is much more accurate than something somatosensory like the perception of pain associated with electric shock. And here we see the Stevens power function expressed as the Stevens power law. And you can see that the misperception of the stimuli is given characteristic names. So the apparent length discriminations are accurate. The brightness represents what's referred to as response compression, where in a sense as the light gets brighter, we don't perceive it as much brighter. So notice that a brightness level of 90 is perceived at around a little over 40. A brightness level of 80 is perceived a little over 40, and so on. So the numbers are compressed in the sense that high levels of stimulus intensity indicate relatively low levels of magnitude estimation. Expansion is the electric shock example, where the stimulus intensity of a 40 is perceived as, off the scale as an 80. A 30 is perceived, a 30 isn't perceived too bad, but a 50 is perceived as an 80, a 60 perceived as an 80 or more. It's sort of like so misperceived that stimulus intensity is ignored after level 40. It's just all a god-awful stimulus. Well, anyway, these power functions, you see, are given a function with an exponent. So you have the constant is a function of the stimulus with some exponent, and that gives you that curvilinear shape to the functions. So if it's squared, it looks like electric shock. That looks like, y equals x squared. But at any rate, the power function refers to the stimulus intensity being raised by a certain power, and that gives you a curvilinear relationship between the stimulus intensity and how we perceive its magnitude. Now we have to have a general idea of what receptors are, especially in their relationship to neurons. Now, receptors are a special device of evolution that's used to detect these things in sensory systems. And here we have an example of rods and cones in vision. But then there's other examples of the cilia cells, the hair cells, and hearing. Then there are specialized receptors in the skin for touch. There are taste buds for taste. And there's essentially smell bulbs for smell. And so you have the receptor, and then you have the sensory system that it's in. And here we have an example of the retina, and I'll be going into this in much more detail in later sections. But here you see the relationship between the receptors and parts of the nervous system that are involved in the perception of vision. The bottom part of the retina, that blue, sort of blue-purplish part down there, is the layer of rods and cones. And you see them roughly designated, with the rods being cylindrical shape, the cones being sort of conical shape. The rods respond to light luminance, to light-dark levels. The cones respond to light frequency and enable us to construct color. We can see how they're all connected. The rods and cones that connect to a ganglion level there in the middle, in which there's some basic processing. So there's an inner nuclear layer here, an inner plexiform layer, and then finally there's a ganglion cell layer at the top there that represents the neurons that get information from the layers below. They bundle up and form the optic nerve that goes back into the brain. So the neurons have a relationship to receptors in which they're going to be stimulated in characteristic ways depending upon what the stimulus is. And the pattern of this conversion is still largely unknown. It's known there's obviously a pattern, but the pattern of firing, how that's conveyed to the nervous system, is largely unknown what the patterns actually are. Some theories are that it's just level of firing, places of firing. Anyway, there's a lot of different theories that try to explain this, but it's still a lot more research needs to be done to figure out how the receptors encode the information and how they transmit that to the neurons, and then how the neurons convey that up into the central nervous system where it's processed. Here's the basic structure of a neuron. You may have seen this before, especially if you've taken physiological psychology or any biology course that included a discussion of what neurons are. But I need to go over it with you just so you have a basic overview, because I'm going to be referring to different parts of the neuron, how neurons function generally, and all through the course, because the central nervous system is connected to all these sensory systems. And so all the basic terminology I just had to very briefly go over here. Well, here's a generic picture of a neuron with the major component parts of one. You can obviously see there's a cell body there on the left. The cell body consists of a nucleus and the structures generally of cells. There's an axon, which is a stem coming out of the cell body, and it terminates or ends at what's referred to as terminal buttons. There's also on the cell body here depicted dendrites. Dendrites are parts of the membrane that are extended out because axons from other cells will stimulate dendrites and also will have terminal buttons right onto the cell body of other neurons. So other neurons are connecting to the dendrites. They're firing across, and they're stimulating this cell. This cell gets stimulated enough, it fires, and it fires down the direction that you see the arrows going. All neurons fire from the cell body down the axon to the next, to the terminal buttons. Now you can imagine another cell connected to this cell at the terminal buttons. So you have other cells coming in the dendrites with their terminal buttons from their axons to the dendrites of this cell, causing it to be stimulated to fire. That electrical conductance being carried down the axon to its terminal buttons to the dendrites of another cell. Now also depicted here is the myelin sheath that covers the axons. In this myelin sheath is special cells that wrap around the neuron. In this sheath, this covering facilitates the conduction of the nerve impulse. Okay, so neurons basically consist of dendrites, cell bodies, and axons, and they're all part of a communication system in which the cells communicate with each other using a one-way transmission of a neural impulse that goes from the cell body down the axon to the next cell. A neuron has a special membrane. It has evolved over the course of millions of years so this membrane is able to conduct a neural impulse. Now all cells have a membrane characteristics that have the chemical properties to produce impulses, and I presume some of them actually do produce what would be considered an impulse from time to time. But neurons have specialized in order to have these impulses as their major function. So neurons are unique because their functionality is not defined by their structure, like say a kidney cell would be or a blood cell would be. The neuron's function is defined by what network of neurons it's among. So if it's among the motor neurons, then it's responsible for motor conduction, that you move and so on. If it's part of a sensory system, then it's part of a sensory system, and you're able to use it to hear better, see better, things like that. The neuron itself doesn't, of course, know where it is, what system that it's in, but its function is defined by what system it's part of, and not by its structure. All neurons more or less work the same way. It involves the small exchanges of potassium and sodium across the membrane, causing a very, very tiny discharge of electrical ionic discharge as these ions cross in and out of the membrane. It produces a pulse that when you control the pattern of pulses, then you've got information. So if you control the pattern of impulses, then you've got something that's going to be sending hearing information into the central nervous system. Now all neurons have impulses like this. Some of them are tailored for different kinds of receptors and things like that. It can get very, very complicated. But when it comes down to it, they're still basically the same. They have receptor sites for certain stimulating compounds. These cause depolarizations of their membranes. That's the neural impulse that travels down the axon. And this neural impulse may stimulate other neurons. And that's basically how the system works. Now what's going to happen in sensory systems, which is part of this course, is you're going to have neurons connected to receptors. And so a receptor is going to do something to depolarize the neuron so that it fires. And the receptor does this consistent with the kind of stimulation that's out there. And the firing patterns that are generated by the receptor stimulating the neurons end up being perceived as certain sensations by the central nervous system. Here you see a graphic that depicts the flow of ions across the membrane and how that affects the action potential. It's also depicting the voltage change that occurs across the membrane. You can see it's a very, very small little change, but you don't need much. You don't need not like you got to, crank up the wattage here. You don't need much power. All you need is a little bit of exchange of power, a little bit of voltage change, so that the impulse occurs and the firing rate is perceived in the central nervous system. This is not unlike the lower power levels of personal computers. You don't need a lot of power with computers to process things. What you need is a systematic system of voltage regulation so that information is transferred within the personal computer. The only thing that really requires power in the computer are things like the hard drives and the screen and things like that. The actual processors do require power, but it's pretty low level. Well, your nervous system doesn't require much power either. You just have to have enough to produce reliable pulses. The pulses are going to communicate the status of, say, its sensory system, and that information, as long as it's conveyed in a way that the pulses are reliable, that's all you need. So here you see the action potential being created by the opening of sodium channels. There's a change across the membrane of a few levels of voltage. Then there's recovery as the membrane comes back online and reaches its threshold, it reaches its baseline state and gets ready for another pulse. When it's depicted like this, it looks like it takes a long time for a neuron to produce a pulse, produce an action potential. This happens within just a millisecond or so. So when these neurons are firing, they can fire like a machine gun. Just very rapidly, they can produce a series of impulses that convey information. Then they can stop, produce more impulses, and the firing rates and the patterns that are associated with those convey information about the underlying sensory condition. So when I press down on my skin and I feel that, there's a whole group of neurons there that are depolarizing and sending impulses up into the brain. My brain then interprets that information. I localize where I'm pressing down, and I can even control the pressure based upon my motor system. It's also involved, obviously, in putting pressure on something. But at any rate, the patterns of firing, the patterns of action potentials from groups of neurons associated with sensory systems are used to convey information by firing patterns up into the central nervous system, where the firing patterns are interpreted as sensations and perceptions. The final element to review is the synapse. The synapse represents the connection between two neurons. And here you see it depicted in this graphic. The neuron at the top, what you're seeing there is the terminal bud of the axon. And presumably at the bottom, you're seeing a connecting section of a dendrite or the cell body. And this terminal bud has come right up to it and formed a kind of impression into it, and they've accommodated to each other. That represents this chemical connection between two neural cells. Now what happens at the synapse is that the stimulation of the axonal discharge in the cell at the top, in its axon, causes vesicles of neurotransmitter to be released by the cell, be released across the membrane. And you see that in the graphics down below. What happens is these neurotransmitters, when they're secreted across the gap of the synapse, and they're received by receptor sites in the postsynaptic cell, that opens channels in that cell and causes it to depolarize. So it causes an action potential in the following cell. So an action potential in the terminal bud of the axon at the top causes vesicles to open in the membrane. Chemicals are transmitted across the synapse cleft and cause the postsynaptic neuron to fire. Now this is a very complicated process that I've described in very simple terms. There are probably hundreds of different kinds of neurotransmitters. There are probably as many different kinds of receptors. Obviously, there are different kinds. There has to be the same number of receptors as there are neurotransmitters. Now, systems, however, will tend to use their own neurotransmitters. For example, the motor system that's affected in Parkinson's disease involves the neurotransmitter dopamine. And the medication treatments for it involve boosting up dopamine levels. Dopamine is a neurotransmitter in that system. Excitatory amino acids are a common neurotransmitter in the cortex. And acetylcholine is a common transmitter in the muscle system, in the motor system. And what that means is that's the preferred chemical substance across the synapse because the postsynaptic neuron has receptors for it. And you see a receptor down below opening up, channels opening up. that allow for the depolarization of the follow-up neuron. Now, these synapses, in the way they affect the postsynaptic neuron, can involve multiple axonal buds from the same cell. It can involve axonal buds from other cells, and it forms this network. Now, what this a network by which the postsynaptic neuron is stimulated. Now, in general, what this produces is a very strong pulse-oriented system for communication. So in order for this postsynaptic neuron to fire, it's just going to fire all or nothing. It doesn't fire by by levels or by ranges of stimulation. It reaches a certain threshold of stimulation and then it fires. It starts producing pulses consistent with the stimulation it's receiving. The synapse is a kind of a gate, in a sense, on the excitements of the nervous system. The synapse is another control mechanism by which the nervous system is modulated. And the neurotransmitters that are used in it and the complexity of networks that cells are in all combine to produce function. The synapse is also a good model for what the connection often is between the receptors in a sensory system and the neurons they're connected to. A lot of the receptors, what they end up doing is conveying essentially chemical messages to the neuron so it depolarizes, very similar to the way in which a synapse works, where one other neuron is trying to depolarize a follow-up neuron. In sensory systems, you have receptors in that position trying to depolarize a neuron based upon the sensory qualities of the stimulus that they're sensing and perceiving. Once neurons are involved in a sensory system, they fire with rates consistent with how they're being stimulated by receptors. Your brain then takes that information and interprets it as a bright light or a dull light or a red light or a green light. The information is then processed and made into a perception from the firing rates of neurons that are connected to the receptors.